Hello friends, welcome to BMH Learning. In this video, I'm going to talk about fusion proteins. The literal meaning of fusion is made of parts from different sources. So, these are the proteins created through the joining of two or more genes that originally coded for separate proteins. We can say it's a protein consisting of at least two domains that are encoded by separate genes. Now, the translation of these fused genes results in a single or multiple polypeptides with functional properties derived from each of the original proteins. Fusion proteins can be created in vivo as the result of a chromosomal rearrangement. Example for such a fusion protein is the BCL-ABL fusion protein, which causes chronic myelogenous leukemia. Similarly, fusion proteins can also be created in vitro using recombinant DNA techniques. Now, there are essentially two types of fusion proteins. Type 1. Of course, there would be at least two proteins. Both of these proteins have N-terminal as well as C-terminal. These two proteins are connected end-to-end, or in other words, the C-terminal of the first protein is attached to the N-terminal of the second protein. And this attachment is done through a linker, or you can say an adapter. Now, type 2 fusion proteins. So in this type, amino acids from both donors are interspersed in the fusion protein product. In simple words, parts of one protein are placed at various positions of the second protein. Now let's talk about fusion proteins as recombinant proteins. Virtually almost all recombinant proteins are now prepared using fusion domains, also known as tags, or we can call them as protein tags. So what is a protein tag? Protein tag refers to the subdomain or the peptide sequence of a fusion protein. There are many kinds of protein tags which are widely used in recombinant protein production. The most important use of protein tag is purification of recombinant proteins. So fusion tags are used as affinity tags. That is, the recombinant protein to be produced is attached with a fusion tag. The tagged protein can be easily and conveniently purified by affinity chromatography. Now, there are a number of fusion tags which help in the purification of the recombinant protein. These tags may contain Staphylococcus protein A, glutathione S transferase. Sometimes maltose binding protein is used. Similarly, cellulose binding protein is a good option. Then we have very popular example of polyhistidine tag, and the list goes on. Another main application of fusion tags linked with the recombinant proteins is to enhance their solubility. Many recombinant proteins are produced with poor solubility, so fusion tags are used to increase their solubility. Fusion tags can also be used as reporters to track the expression level of the specific protein that the researchers wish to study. Other than this, the protein tags are also used to simplify protein refolding and increase its efficiency. Among the fusion proteins, one of the most widely used categories is called fluorescent proteins. These are green fluorescent protein, orange fluorescent protein, and yellow fluorescent protein. These fluorescent proteins enable direct observation of dynamic intracellular processes. This was all. Thanks for watching.